Hello everyone, this is Dr. Tushar Dutta. I'm working as a scientist in the Indian Agricultural Research Institute, New Delhi. The topic of my presentation is the potential of Photorhabdus bacterial toxins for usage as bioprotectants to manage crop pests. Entomopathogenic nematodes are soil inhabiting organisms. They are obligate insect parasites. Heterorhabditis nematodes symbiotically associates with Photorhabdus bacteria. Steinernema nematodes symbiotically associates with Xenorhabdus bacteria. And this bacterium is harbored in the intestine of infective juveniles. EPNs have been extensively used as a biocontrol agent of different insect pests. This is the life cycle of EPN. Infective juveniles enter the host by natural opening or rupturing the cuticle. They release the bacteria. Bacteria produces toxins and kill the insect within 24 to 48 hours. And nematodes start development by feeding on the dead insect tissues. They reproduce and then infective juveniles reassociate with the bacteria. Then they emerge out of the old host to search for a new host. The efficacy of native bacterium or live EPN as biocontrol agent is very much limited because of their narrow range of soil moisture and temperature requirement, and their shelf life is very poor. So the attempts are made to reproduce their insecticidal activity of Photorhabdus in different microorganisms, because Photorhabdus is a gold mine for discovery of novel insecticidal toxins which can be exploited by transgenic mean or biopesticide formulation. Majority of Photorhabdus luminescence toxins are insecticidal by injection, which is not very amenable for future commercial exploitation. In view of this, we started to explore the orally active toxins from Indian strains of Photorhabdus acharstei. We isolated 6 strains from different geographical locations. We characterized a number of toxins from these strains. For example, TXP40 toxin was isolated from different strains which showed injectable toxicity and oral toxicity to Galleria melonella. And we seen that strain specific sequence divergence was correlated with their differential virulence against Galleria melonella. Then we characterized a 63 kilodalton protein TCAB, which contains a zinc metalloprotease activity domain, which is directly involved in bacterial virulence. And interestingly, a homologue was identified in Bacillus thuringiensis cryotoxin domain. It showed injectable and oral toxicity to Galleria melonella, and again their differential strain virulence was correlated with their different sequence divergence in the amino acid level. Then we showed that TCAB toxin is very much cytotoxic against the Galleria melonella hemocytes. We then again characterized a few of the more toxins such as SPEED-A, TCAA, TCCA, TCCC. Then we identified a new Photox class toxins, Galtox, which is around 35 kilodalton in molecular weight. Then we established the efficacy of these toxins in Helicobacter parmigerus, Sporoptera litura, and Sporoptera exigua. We showed that these toxins can interact with the midgut receptor proteins such as caserin, APN1, ABCC, and ALP. And in the process, they create a pore-like formation in the midgut. They rupture the midgut epithelial cells and then travel to the hemocoel. The stability of the protein was also established in room temperature. Then using force feeding assay showed that the toxin gradually travels from the midgut to the hemocoel, and we showed that the midgut epithelial cells are badly ruptured and they create a pore-like formation in the basement membrane and visceral muscle layer, which is alike of the Bt toxin mode of action. This mode of action was also established in Spodoptera frugipoda, Helicobacter parmigerus, Sporoptera litura. We conclude that due to their smaller molecular weight, these toxins are amenable for deployment in insect-resistant transgenic crops. Earlier, a TCDA toxins from Photorhabdus luminescens was expressed in Arabidopsis, which showed resistance against Manduca sexta. A recent paper also showed that TC holotoxin can be formulated as biopesticide molecule. The major concern is now that the insects are showing resistance against the cryotoxins. Therefore, if the Photorhabdus toxins can be adopted as an alternative, it can strengthen the pest management repository. Thank you.
Hello everyone, my name is Harpreet Kaur. I'm a graduate student in Department of Plant Sciences. Today I'm going to talk about cover crops' impact on water quality and crop yield in a tile-terraced no-till field. As cover crops has been researched in Midwestern states and found to be a management practice to reduce nutrient losses in surface runoff. Whereas Terrace construction can also be a helpful tool in reducing soil erosion. Thus, combining these two properties could have a synergistic effect on nutrient losses in water. Therefore, to study this effect, experiment was initiated in 2016 with construction of terrace in each plot, and each plot was individually tile drained. These 6 plots received 2 treatments, cover crop and no cover crop, through randomized complete block design. We used 2 kinds of cover crops in this experiment. One was the cover crop mix, which includes wheat, radish, and turnip, and that was aerially seeded into soybean at R6 stage. And the other was cereal rye, which was drilled after harvesting corn. As you see in this picture, each tile drain line from the individual plot passed through these trapezoidal flumes, where we have automated water samplers, and those water samples were analyzed for total suspended solids, nitrate, total phosphorus, and dissolved active phosphorus. Here is the event mean load values for these nutrients from each season. Firstly, talking about cover crop season 2018-19, where we planted cover crop mix as wheat, radish, and turnip. We found a significant reduction in total suspended solids and nitrate from cover crop plots compared to the no cover crop plots. This reduction could be because as cover crop canopy reduces the kinetic energy of raindrops, thus reducing the splash effect of a raindrop. Also, cover crops uptake nutrients from the soil, therefore reducing the nutrient availability for water losses. While in corn 2019, we found a significant increase in dissolved active phosphorus from cover crop plots compared to no cover crop plots. As cover crops mineralize, there might be the release of the nutrients into the soil. And in addition, the study was in a no-till. Therefore, there might be the chances that the phosphorus stratification might have occurred in the upper 6 inches of the soil, due to which more phosphorus was available for water losses. And in 2019-20 cover crop season, where we had cereal rye, we didn't observe any changes in these nutrient values from cover crop plots to compared to no cover crop plots, which might be because as early in the season, because of temperate climate, we don't get much cover crop established. While later in the season, we applied fertilizers, and that might have diminished the effect of cover crops. Also in the soybean season 2020, I would like to mention here, this data was not from the whole soybean season. This was like up to July 2020, where we found a significant reduction in total suspended solids and nitrate. This reduction might be because of there might be a cover crop residue laying on the ground, which might have reduced soil erosion and the nutrient losses. Talking about the crop yield, we found a significant higher cover crop biomass in cover crop plots compared to no CC plots, which was expected as we are having a treatment over there and the other was not treated. And we didn't found any effect of cover crops on cash crop yield. Whereas we had a significant effect of topography on yield. As you can see, we had a lower yield in channel positions, which was because in channel we don't get much crop establishment because of the weather conditions. In summary, we found cover crops in no-till tile-terraced field reduced nutrient loss without affecting cash crop yields. Also, the impact of landscape position on yield is an important factor when we consider terrace construction from a farmer's point of view. Thank you so much for your time and attention. I will be happy to answer any questions.
Hello, my name is Chenhao. I'm from Nanjing Agricultural University and working with Dr. Bing Yang at MU. My research focuses on the interaction between rice and the pathogen XO. Here we would like to use CRISPR-Cas9-based genome editing technology to improve the rice resistance. As we know, bacterial blight of rice is a severe bacterial disease on rice. The capture agent XOO could secrete tear effectors into the plant cell. Tear effector directly binding the sweet gene promoter induce its expression. The high-level expression of sweet functional exporting sucrose out of the plant cell. Then the pathogen can use those sucrose for the further development and multiplication. If so many pathogen bacteria around the plant cell, the rice will not be happy because the disease happens. And TLC as an essential valence tear effector, which was found distribution around all the sequenced African XO strain. But there is no natural rice resistant resource was found. To our knowledge, the valence of TLC mostly depend on the induction of sweet 14. So we try to apply the CRISPR-Cas9 based single base pair editing technology. Here is rice CBE, cytosine base editor, and ABE, adenosine base editor, to modify the sweet 14 promoter domain. The TLC target sequence. After rice transformation, we successfully got the desired mutant 2C changed to 1T, 1G, or 2C changed to 2T, and 1A changed to 1G, and another A changed to G. As you can see, 2C modified plants show fully resistance to TLC, but single A modified plants has not enough to resistance TLC. In the 2C modified plants, the sweet 14 gene could not be induced by TLC, but in the single A modified plants, the sweet 14 gene still can be induced, which matches the disease phenotype. Also, we measure the major traits of the mutants. We can see here the speculate and the seed size similar as the wild type. So the conclusion here would be 2C modified plants show completely resistant to TLC and without the yield penalty. Also, the base editing technology would be the powerful biotechnology for the crops improvement. In the end, I would thank my supervisor, Dr. Bing Yang, and all the lab members. Also, thanks for the foundation support. Thanks for your watching.
Hello everyone, I'm Gourav Sharma. I'm a PhD candidate in weed science at Virginia Tech. Today I'm going to talk about understanding weed evolution through epigenetic lens. As we all know, population is increasing and by 2050 we will be around 10 billion people. In order to feed everyone, we have to increase our food production by more than 50% of what we are producing today. But in order to achieve that goal, insect, pest, disease, and weeds are the major obstacles. Weeds are the unwanted plants in the field which cause yield loss to farmers and food loss for all of us. Recently done study in corn have shown that if farmer does not control weeds in their farm, it can cost a loss of up to $26.7 billion annually. So what farmers do, they spray chemicals known as herbicide to kill those weeds. However, due to the overuse of the same chemical, these weeds now becoming resistant to the herbicide. This is similar in human, we known as antibiotic resistance, where we are not able to control the bacteria with the same medication what we were having for the last 10 years. If we talk about the resistant cases, we have seen some severe cases in Missouri and Arkansas where farmers lost their whole crop due to the resistant weeds in their farm. They were not able to control the weeds with the same chemical they were using for last 10 to 15 years. Scientists from the past 2 decades, they are trying to understand why these weeds are becoming resistant. But however, nobody have looked from the epigenetics point of view. And one such example to understand the role of epigenetics is the case in identical twins. Have you ever imagined why two identical twins originating from the same genetic material, yet one of them develop a heart disease later in the life and other does not? One reason could be epigenetics. So think this way. At every construction site, there is a blueprint. And on that blueprint, there are sticky notes, which have instruction for worker, which tell them what to do and what not to do. Similarly, DNA is the blueprint of plant cell. And on that DNA, there are some sticky notes which tell plant what to do and what not to do. And the most common sticky notes we study is the methylation. And to understand the role of herbicide resistance, we are working with sorghum bicolor since it's closely related to weeds such as Johnson grass and sugarcane, which are big problem in US and Canada. So we grow our plants in the greenhouse. We give them two herbicides. These are the most commonly used herbicide in the ag world. And we also give them two other stresses, clipping and shading, to mimic what happened in the nature. So clipping resonates to the insect herbivory, where insect eat the plants, and shading is when weeds are growing under the crop canopy. We collect the leaf for the DNA extraction and further analysis. And what we have found that the plants which were treated with the glyphosate, they had lower methylation, meaning there is a removal of the sticky notes from the blueprint. And when those sticky notes are being removed from the blueprint, it's causing some genes or some other part of the blueprint to express more, which is helping plants to adapt to glyphosate and develop resistance. So we have seen that the removal of the sticky notes causing herbicide resistance, and we can use this information to develop new herbicides which will be less prone to develop resistance. And also we encourage our farmers to use multiple different herbicides. Do not rely just on herbicide to control weeds, instead use integrated weed management approach. And with that, I would be happy to take any questions. Thank you.
Hello all, I am Deepika, master student from India. I welcome you all for MU Plant Symposium 2021. Today I am going to present the topic induced parthenogenesis and haploid regeneration in papaya. As you all know, papaya is one of the most economically important fruit having a global demand. It's a very delicious tropical fruit known for its high nutritional benefits. Papaya is usually propagated by seed, and papaya ring spot virus is a major problem for better yield and quality. Apomixis is a natural method of asexual reproduction in plants in which seed development takes place without fertilization. Apomixis can be exploited for breeding programs by producing double haploid plants through induced parthenogenesis. The major advantages of double haploids are there will be no segregation of genetic traits in the population, and there will be shortening of breeding cycle, and the generation of homozygous breeding material is possible through double haploids, and the gynogenesis will overcome the disadvantages associated with anther culture. So, using modern biotechnological tools, tissue culture can be used as a potential strategy for overcoming problems of lack of efficient breeding material for cultivar improvement. The materials and methods we have used are we have collected pollen grains across the field from hermaphrodite papaya plants. We have irradiated these pollen grains using gamma rays of 0, 25, 50, 75, and 100 grades followed by pollination. After that, we harvested the fruits of 30 and 45 days of pollination, and we aseptically cultured the ovules developed inside the fruits in MS media with different hormones. Research suggested that the number of ovules and size of the ovules decreased with increasing concentration of gamma rays. In figure 1, you can see the average number of ovules present in 30 days and 45 days. The average number of ovules was more in 45 days fruits compared to 30 days fruits. Also in plate 1, you can compare the size of the fruit developed after 30 days after pollination and 45 days after pollination. In figure 2, you can see the percentage of callus from ovules of 25 grapefruit. The regeneration of callus from the unfertilized ovules in vitro was recorded highest in gamma irradiation of 25 gray in treatment T14, that is MS media with NAA of 0.2 mg per liter with BAP of 1 mg per liter the 30 days explant. In overall result, the 30 days explant has given the good response when compared to 45 days fruit. So in plate 2, you can see the shoot induction from unfertilized ovules after 6 months of subculturing. And in plate 3, you can see different types of callus obtained from ovules by using different combinations of growth hormones. And also the supplementary observations like multiple embryos you can see in this image which is taken under microscope. And the callus regeneration, also the shoot regeneration from indirect embryogenesis and with direct embryogenesis. The results concluded that the pollen irradiation method is one of the best methods and the better technique to induce parthenogenesis in croplands. The media combination of NAA and BAP is best suitable for callus induction from ovule explants in papaya. Double haploid production in fruit crops will reduce the time required for breeding purpose, which is a major goal of the research. Further studies on direct regeneration of roots and shoots from ovule explant is necessary. Finally, I would like to thank MU Plant Research Symposium for giving me a great opportunity to share my research and the results worldwide. Thank you.